Thank you very much, everybody, for joining this GetGenome seminar. My name is Dr. James Canham, and it's a real pleasure to introduce Ben Pascoe and Evangelos Mourkas. First of all, Ben is a senior research fellow at the INEOS Oxford Institute for Antimicrobial Research and the Department of Biology at the University of Oxford. His research focuses on the genomic epidemiology and evolution of bacterial pathogens, with a particular emphasis on foodborne zoonosis and antimicrobial resistance. And I think I'll introduce Evan a little bit later. So we'll let you start, Ben, and thank you very much for presenting this GetGenome seminar.
Well, thanks for inviting us along, James. And yes, so I'm Ben. I'm from the University of Oxford. We're going to do a little bit of a tag team effort, and I'm going to try and give a bit of a global overview, and then Evangelos is going to talk a little bit more specifically about some of the work we've done in Peru on an organism called Campylobacter. So Campylobacter is the most common bacterial cause of gastroenteritis worldwide, and particularly in the UK, where in the UK the cases of infection with Campylobacter is more than the combined numbers for E. coli and Salmonella and Listeria. It's recognized by the US CDC as a priority pathogen, and the WHO, it's on, or used to be on, their list of pathogens for which new antimicrobials are needed because there's problems with antimicrobial resistance. And two main species are responsible for disease, particularly in sort of high-income countries where about 90% of infections come from Campylobacter jejuni, and then the remaining 10% or so from C. coli. And so in the UK, this is something from an FSA study a few years ago where they went out into different supermarkets and swabbed chicken from all the different suppliers. And you can see the numbers are all pretty high from all the different supermarkets. So Campylobacter is like having a Cat2 pathogen in your kitchen every day, because the main source of infection for humans, at least, is through eating sort of contaminated, undercooked meats, and particularly chicken in high-income countries, at least anyway. And some of the work that me and Evangelos do is to sort of understand differences within Campylobacter, because not all Campylobacter are the same. In most cases, you would eat your chicken, you might get ill, and you'd have a bad stomach for a week, but after a week, most people would get better and the infection would resolve itself. And in Britain, we wouldn't give most people antibiotics to do that. It would do it on its own. But sometimes we have these post-infection sequelae, such as post-infection IBS, and this is some work we did a few years ago looking at different Campylobacter from human cases, and we had follow-up data about the patients and which ones developed IBS and which ones didn't. And you can see traces, or you can see signals within the genomes and markers for sort of severe infections or development of these post-infection complications in specific lineages. And in this case, this was work with Madhu Grover from Mayo Clinic in Minnesota, where we had this great follow-up data, because usually we don't have that kind of information. And then we've done a similar thing with Shaoqi and in China, where they have this follow-up data. And in this instance is Guillain-Barré syndrome. So that's a really nasty post-infection complication where you get where the body attacks the Campylobacter, it removes the disease, but then it keeps attacking itself, and you get these neuropathies. And Guillain-Barré syndrome is really nasty. And when we look at the isolates that cause Guillain-Barré syndrome, we can see clustering within specific lineages. And if we make trees of these genomes, we can see that they actually cluster in specific parts of the tree. And these are often from reservoirs that are in mammal origins rather than chickeny type ones. And this is how we can detect these signals within the genome that we think predict the the disease severity and whether people are going to develop complications from disease. And we've seen recently that this has been a problem around the world, and there was a big recent outbreak in India of Guillain-Barré syndrome linked to Campylobacter infections that people picked up from drinking dirty water. And this was a particularly large and nasty outbreak where there were several people died and lots of people became ill. And we see these neurology type conditions where people have to go on ventilators and they lose the workings of their limbs. And if we go back to Xiaoqi's work, we can sort of try and understand which Campylobacter cause these type of post-infection disease sequelae by looking at the genomes, looking for markers. And then this is a complicated way of showing the genome. So we have a genome on the top left corner, and it's been chopped up into all these different small pieces. All these dots are individual chimeras. And then we can do some statistics to look at which genes or which chimeras are more often found in disease cases or severe disease cases, and identify these genes and identify specific alleles of these genes so that we can try and predict disease outcomes from genomes and then provide adequate advice and healthcare treatments. And this is essentially the kind of approach that Evangelos and I have used in different Campylobacter populations around the world and in lots of our work. And because of this strong host signal that we see in Campylobacter, we can do source attribution studies. And the way I like to think about source attribution studies is to remember this scene from Forrest Gump. So Forrest Gump is sat on the bench waiting for his bus, and the nurse comes and sits next to him and he says, there's an awful lot you can tell about a person by the shoes that they're wearing. So the nurse is wearing a uniform and she wears different outfit, and from that outfit you know where she's going or where she's come from, and you can predict what the work that she does. So do Campylobacter have different shoes? Can we predict these disease outcomes from the shoes that they wear, the genes that they carry? And that's what we that's what we try to do. And we looked into a large data set and did this on a large scale with all of the genomes that America had sequenced over the last 20 odd years. And we tried to predict where people had become infected, the foods they might have eaten to pick up their Campylobacter infections. And you can see the strong host signal in Campylobacter from the trees. So each leaf on the tree, each dot is colored by the source that it's come from, and you can see that the colors bunch together, particularly in C. coli. And then our human cases you can see that if we put our human case onto a tree, you can either predict with confidence where that person might have picked up their infection, or even in some of the messier parts of the trees where the colors overlap, we can put a number on that. We can give it a probability. And we use some complicated machine learning tools to try and identify specific parts of the genome where they segregate quite well between sources. So this is this complicated plot on the top left, where we see parts of the genome segregating clearly between chicken, cattle, and wild bird sources. We use these markers then to predict where people might have become infected. And in America, at least 70% of infections we thought came from poultry, from eating contaminated or undercooked chicken. And we did that on a regional basis as well. So you can see that on the maps, there's slight differences in coloring. So there's in different parts of the country, in parts of America where there's a lot of chicken farming, there were more cases attributed to infection from chicken sources. And this is consistent with what we've seen around the world when we've done this in other countries. So in France, we tend to see a few more infections from beef and cattle sources. We see when we compare between and within countries, we see that within a country is more similar to between countries. And generally, for Campylobacter at least, everywhere we look, poultry is the #1 source, but we see slight differences between cases. And this sort of idea of a global perspective on Campylobacter research and not treating each Campylobacter the same and then not treating each country the same is consistent with what we see when we look in public repositories. So whenever you sequence a genome, you make it public so you can publish your paper. And this is all the data from PubMLST, which is where Campylobacter researchers would tend to submit their data. And we see some lineages that we find in all around the world, in all 6 continents where we have isolates from, but in most cases, we see these sort of local strains, local lineages that we only ever find in specific places. So we have both locally restricted lineages plus these nasty global circulating isolates that are probably spread by the poultry food chain. And this is this global perspective on Campylobacter research is what we bring to the work we've done. And then Evangelos now is going to talk a little bit about specific work that we've done in Peru and how we've used these genomic techniques to understand what's going on locally in Iquitos. So now I'm going to hand over to Evangelos.
Evangelos Mourkas is a researcher at the Zoonosis Science Center, Uppsala University in Sweden. His research focuses on the evolution, transmission, and antimicrobial resistance of zoonotic bacterial pathogens, particularly Campylobacter. Using comparative genomics and machine learning, he investigates the ecological and evolutionary dynamics driving pathogen emergence. So I'll hand over to Evan. Thank you.
Thank you, James, for the introduction and for the invitation. So let's check that my setup is fine and you can see my screen, not the presenter mode.
All good.
Okay, thank you. So yeah, Ben has done a brilliant presentation introducing global differences. So I'm going to share some insights, genomic insights, but also infection insights and the evolution of AMR in Peru. So this is part of an NIH grant, which is led by Margaret Kosek at the University of Virginia, and together with Francesca and the team in Iquitos, they've done some brilliant sampling of newborn children on the edge of the Amazon in Peru. So it's located here, where they routinely sampling of newborns every few months. And they've been following taking samples when they have or when they haven't got any symptoms of diarrhea. So they have done a lot of sampling in the ground, and we have looked at the genomes together with Kerry Cooper at the University of Arizona and Craig Parker at USDA. And more recently, Madison from University of Oxford has joined to the team to study some of the metagenomic samples that we have from this sampling. So we have consistent level of identification of Campylobacter from these children, as I said, even in the absence of symptoms, so diarrhea. So this is how the lab looks like. So it has all the basic equipment that you need to grow bacteria and do basic molecular microbiological analysis. And it's located in this beautiful building in Iquitos. I haven't had the chance to visit the lab yet, but Ben has been there, and it's really cool what you can do in just a room with the right equipment. So we know from the malnutrition studies the incidence rate of different pathogens, but also of Campylobacter. And we know that the children there experience at least one persistent episode of Campylobacter, with some countries having a higher incident rates than others. And we've seen there that there is a high incidence in Peru. So in the original study, there was a significant low level or carriage of Campylobacter jejuni, coli, or larvae. But the team has improved identification. So they have improved the culturing protocols and the methods and new primers to find Campylobacter, but also separate by the different Campylobacter species. So we know now that the prevalence of asymptomatic carriage is higher even than that. The team in Peru has also identified an association between carrying Campylobacter with weight gain. So in children, so really having Campylobacter associated with growth stunting, which is quite bad. And because the team are routinely sampling the same kids over a long period of time, and that is from 2 to 5 years, they're able to identify potential cases of persistent infection. And this is repeated isolation of Campylobacter from the same person over a long period of time, months. And most of these cohorts now are reaching an end, since the funding is reaching an end. So we'll soon have a good set of isolates and genomes to work with a lot of metadata. So we really want to see if it's the same isolate, strain, or lineage that's causing these episodes, and is it really a repeated infection due to high exposure rates? So is it really a chronic infection, or is it repeated infection from the same, from having access to the same source, let's say water, or is there even something else like human-to-human transmission within the families? So as Ben already discussed, we know a lot of what's happening in high income countries, but we don't really know much on the situation on transmission dynamics in low middle income countries where exposure is much higher. And then bringing really the use of antibiotics in these low resource settings, one can go to the counter and buy whichever antibiotics they use. So there's really unrestricted use in that. And I think, if I'm not mistaken, these are photographs that Ben has taken in Peru, and I have similar photos from the Gambia and in Bangladesh.
Yes.
So we wanted to firstly check what is the situation with our isolates. So looking at our phenotypic and genotypic resistance of the collection that we have, we see that there is extensive drug resistance in these Peruvian pediatric isolates, and we see that there is much more resistance in coli compared to jejuni, and we see that both in silico in the genome, but also phenotypically. And we have seen that before, so it doesn't come as a surprise. But what is a surprise is that there is really high levels of resistance against ciprofloxacin and azithromycin, at least for Campylobacter coli, which are the two oral antibiotics that they are used to treat Campylobacter in Peru. So that really leaves us no option or easy option to treat Campylobacter in these settings, because then the other treatment option would be the use of antibiotics IV, so in the blood, and that's not possible in these settings. So this looks like a complicated plot, but this is something that we always like to put when we have a lot of metadata. So we want to compare all these genetic determinants that we see in the genome and see how they correlate with the phenotypic resistance that we see in the lab. And then we see that for a lot of the antibiotics, we sort of have like a good explanation of, oh, we have resistance in the phenotype in this antibiotic because that SNP or that gene is present, but it's not always the case for all of the antibiotics in our collection. And then a lot of people that use genomics know that you can screen your genomes for the presence of all these antimicrobial genes, so these genetic determinants, using publicly available databases, and AMR Finder Plus is one of them. But these databases have been validating using different datasets. And then in the case of azithromycin, they report 100% consistency, but in their training dataset, they had no isolate that was resistant against azithromycin. So one needs to take that under consideration when things don't make sense when you screen your isolate and your genomes. So you need to know the tool you're using and what it has and how it has been benchmarked. So then we started to dig in the genome and trying to understand what explains the phenotypic resistance in the proportion of our isolates. And we saw different combinations of CMEB alleles, and that is one of the genes in the efflux pumps in Campylobacter that has been associated with resistance before, but also some promoter regions with enhanced activity, which confer broad spectrum resistance. So we have high incidence in this population of Campylobacter infection, and also we have asymptomatic carriers. And chicken is likely the primary source. We can see in the photo that Ben, I think, as well, has taken a man buying his chicken from the chicken market. You can pretty much understand that this is a very high level of exposure for the population to Campylobacter. Okay, so we have identified these high resistance CMEB genotypes. So we wanted to look where it is in our genome collection. So we have multiple lineages isolated, and we also have multiple sources where we have isolated Campylobacter. And we see that it's not really restricted to a specific lineage or a specific species, but it's pretty much dispersed among different Campylobacter lineages, and it's found both in Campylobacter jejuni and also in Campylobacter coli. And this is basically the two phylogenies, the jejuni one on top and Campylobacter coli at the bottom, highlighting what I just said, that we really have dispersed phenotype. So it's not a restricted lineage associated with a single source or that has the high resistant phenotype of CMEB. What's worth of noting here is that in Campylobacter coli, we have these two different clusters, so to say, which correspond to two different lineages. And this one here is of particular interest, 1150, because it's not very common in the developed world. And if I'm not mistaken, what we have now in the public database in PubMLST in terms of this clonal complex is heavily coming from the Peruvian collection. So it's very interesting, and Ben has been working on this. So yes, we wanted to dig in and understand the evolution of CMEB compared to the other genes that together they make this multi-drug, or they make this efflux pump. So we wanted to understand what's special with CMEB and what does it pop up in our analysis. And I'm not going to bore you and get into very much detail on the figures, but really simply we wanted to compare the predicted protein content using a principal component analysis to see if there is any clustering by species or any clustering by presence of the allele, and we couldn't really see anything into that. So then to understand a bit better, we got the different genes separately, and we looked at so we calculated what we call the consistency index. So if you look at each gene separately, how well does it follow the evolution of the other genes that you have in the genome? So really, what we are after is how much pressure we have in the different genes, evolutionary speaking. And then we made single gene trees for every single one of these genes, and we saw that let's start by explaining a bit what the gene do. So CMEA is the one that forms the periplasmic subunit of the pump, and in this case, when you do a single gene tree, we see that isolates almost completely clustered by the different species, with small subclusters, isolates from poultry and then from human sources. CMEB is the inner membrane subunit, and the protein sequences are segregating almost completely by their source, and that is you can see in the coloring on the tips here, so the orange and the blue. So there is segregation by human or poultry. And then the CMEC is the outer membrane subunit of the efflux pump. And then you can see that when you compare the isolates, we can see segregation also by species, with some mixing of poultry and human clinical isolate. So we wanted to dig in a bit further to analyze the operon itself using structure, which is a software that assigns an individual to source population. So I'm not going to get into the detail of how the model works, but really what we were aiming to do with this was to see where these resistant alleles of CMEB are originating or are coming from. And using this model with that 98.6 accuracy, we saw that they're most likely originating in Campylobacter jejuni. So we wanted to put all of our findings into broader context, and we saw that around 40% of our Peruvian isolates, of the ones that we have sequenced until that time where this project was published, 40% had the enhanced resistant CMEB genotype. But we wanted to see where else can we find this in the world. So we went into our public database, PubMLST, and we looked at all the Campylobacter genomes that are there to see where else we can find that resistant genotype. And this is a good point to note that most of the strains or the available genomes that we have in the database, they are coming from the UK, the US, and New Zealand. And now Peru, I think it has surpassed New Zealand, so it should be number 3. But UK, New Zealand, and US combined, they make around 80% of what there is in the database, which is around 16,000 or 20,000 genomes right now. But what's interesting looking at the map, and you probably already spotted it, is that we see this resistant genotype in high proportions in other low middle income countries, including Egypt and in Vietnam and in other places. So yes, this is also like another way of looking at the data and summarizing where else we can see the resistant phenotype. So we see that it's highly prevalent in isolates coming from chicken. And we see that it has increased over the years, but that can also be more sampling. So keep that in mind. And coming back to the ST1150 Campylobacter coli clone, I'm not going to explain more on this project. Ben is the one that's driving it, but we have a lot of isolates that are assigned to this clone complex in Peru, and we are now starting to understand why, but maybe that can be a separate presentation on its own. So humans are the accidental host of Campylobacter, so it doesn't really live in the human gut. So we have commercial chicken and also chicken meat as the source, as we saw from previously in the photo, which is the same here. But the question is this the source where the population in Peru are getting all of these nasty resistant isolates from? And really, what we see in feces in the farm, is it the same isolates or genomes that survive in the bench here? If they do, which ones are the strains or lineages, and what makes them capable of doing that? So what are the genetic determinants underlying the survival? Because Campylobacter doesn't like to be outside the host, doesn't like oxygen. So in order to survive on the bench in the market, it needs to have specific genetic properties. So there's been some papers from the group dating back to when I started my PhD, where they were trying to understand these functional traits that are linked to Campylobacter surviving from the farm to the fork. And this is a study that's been done in the UK, but also in developing an autogenous vaccine for poultry to reduce these strains that survive well in the environment in the farm level. And this is ongoing work. And now that we have a lot of the data sets wrapping up, we can start then exploring some of this. So this is work that I have been leading in the Peru team. And we see that We have collection of isolates from backyard chicken, so that's feces from these chickens, and then we have commercial chicken, but we also have chicken meat, and that is from the different markets. So we really wanted to see which are the clonal complexes that carry that the chicken meat is assigned for, and we see that in Campylobacter jejuni, at least, the ones that our meat samples are heavily assigned to are clonal complex 353 and clonal complex 607, although there are other small numbers dispersed in the other complexes, but these two are the most dominant ones. And then we did the same thing using the Campylobacter coli population that we have, and there we don't really see a specific trend towards one lineage versus the other, that really the isolates from meat are distributed across the population structure here. And then we wanted to see if there is, what's the genotypic profile of our isolates in terms of AMR. And then we see that isolates from meat, they tend to have more AMR genetic determinants compared to backyard. The commercial is also quite high. And then when we split by the different genetic determinants, we see that for most of them, isolates from chicken meat, they have high resistant levels. And as Ben described, we really like to use the genome-wide association studies whenever we have the amount of the number of isolates and the different phenotypes to do so. So we did a GWAS comparing backyard chicken isolates versus meat, and then commercial chicken isolates versus meat, in order to find these genetic variants associated with survival. But this is in the preliminary stage of analysis, and we see some sort of hotspot in this loci, but we need to control for a lot of different things, including lineage effect and other stuff, which I'm not going to bore you with. So this summarized like a snapshot of 5-year work that has been done and still been doing in Peru. There's a lot of people to thank. This is a multidisciplinary team. The team in Peru, of course, and everybody on the ground in Iquitos, the groups in Oxford, here in Uppsala, but also in other parts of the world. And of course, all the funders. And with this slide here, with some happy moments, maybe we can have some questions.